Okay, now we are recording. So again, exam one, meet me online to get the answers. I mean, technically, I guess we can meet in the office too, whatever. Meet me any way you can to get the correct answers and a 5% boost to your exam one score. Yeah, like I said, you need those answers to study for your final because one half of your final, about 50 questions, is going to come from the first three exams. So the first three exams are your study guide for the final. Any questions about exam one? All right, exam two, you can still retake it. You can retake it until November 9th. You can take it as many times as you want. It's open book, it's not timed. There's no reason your grade should get lower, but even if it does, that doesn't matter. We'll just keep your original higher score. There's nothing to lose. Only catch is you have to let me know when you're done so I can go in and readjust your score. Any questions about exam two? Okay, like I said, exam two retake is closed when exam three is due. And that is on November 9th, right? So exam three is coming up, and it covers chapter 6, 7, 8, and 9. So the stuff we finished today, we should finish chapter 9 today. So meaning, at the end of the day, we are done with the material that's going to be on exam 3. Right, then we're moving on after today, knock on wood, should be moving on to the material for the final exam. So any questions about exam 3? Oh yeah, one more thing again to remind you, you have 50 questions to answer, about 50 minutes to answer about 50 questions. If you need more time than that, let me know. I'll be happy to meet you online and proctor your exam, and then you can take as long as you need to take. So now, are there any questions about exam 3? All right, just to remind you, I'm here to help you for office hours. Today, my office hours from 9 to 12. Those are the times you're guaranteed to find me in my office and online. I'm also happy to meet you anytime outside those hours, but you need to let me know so we can see if we can have our schedules mix and align, which sometimes can be challenging, but anyway, those are my office hours. Any questions about office hours? All right, to remind you, there is a pre-lab due today. It's been a while because we have another virtual lab. This virtual lab is a lot more complicated. So I do plan, currently, you need to be here next week for lab. Even though it's virtual, we're all going to do this virtual experiment together. Therefore, you need to have your pre-lab done. And again, remember, you should prioritize pre-labs over other stuff. Because I will, in this case, I will go back, and even though I have other labs of yours to grade, we'll skip those back there and make sure that you've done your pre-lab. Because I want to make sure you know what you're doing before you come in. The pre-lab only consists of video questions. So, because I just looked at it today, it should have been video questions and hypothesis questions. But I messed up when there was a piece missing of it that said that. So if you have done it, or actually, I guess you could redo it. If you do the hypothesis questions, even though it's not technically required for the pre-lab, I will give you a little bit of extra credit for doing those pre-lab questions. I mean, those extra credit questions. And of course, even though this is a complicated lab, even though we are going to be here to do it, technically, you could go ahead. If you want to try it on your own, you can. And if you turn it in time for me to grade it before we meet for lab next Wednesday at 9 a.m., then then you might not have to come. So you can work ahead if you want to. Any questions about the pre-lab that's due today / next week's lab? Okay. Again, attendance is mandatory. Participation is graded. What time is it? 8:02, good. All right. So at this point, anybody who comes in is late, right? So as always, you need to send me an email. That email needs to have one question about the information presented in this lecture. One question. Even if you think you know everything I said, let me rephrase it. Maybe you could say, even if you know everything, send me a question that would be a good quiz question or a good test question based on the information that was presented in this lecture today. For everybody, that is required. An email with one question about the lecture. If you're in person or online, the other catch, as always, is you need to send it by 9 a.m. The idea is for you to send it right before you leave. Now, if you have an issue with that, like some of you seem to, you can at least tell me why. Maybe I can make some arrangements where you're like, oh, I would, but I have to be at this class on the other end of the campus by 9 a.m. Look, here's my schedule. If you're having an issue that's out of your control, let me know, and maybe we can make some adjustments. Again, as usual, if you're in person or online, you need to include the attendance words or phrases in that email. And of course, if you're watching the video at a later date, you also need to include screenshots every time I say one of those attendance words or phrases. So any questions about any of that? All right, so speaking of attendance, let's get to it. Where are we at here? I accidentally clicked it out. Bear with me, I apologize. Today should be a quick day anyway, so the fact that we're running a little bit behind is no big deal. And again, today should be the last day for information that will be in exam 3. Starting next week, we'll be working on exam 4 stuff. Knock on wood. All right, where you at? There we go. All right, as I call attendance, obviously if you're online and I say you're not here, it's because you're not here. So no need to correct me. If you're online, it doesn't matter. Once you send me the email telling me that you're here at 9 a.m. or before 9 a.m., then you'll be recorded. All right, so Brumfeld is not here. Champion is not here. Gross is not here. Gunther is not here. Randall Hill, both Hills are here. Jackson is not here. Keys, not here. Perez is not here. Sigmund? Yep, Sigmund is here. Slaughter? Huh, Slaughter's not here. Oops. Smith is here. Stover is here. Tompkins is not here. Van Ness is not here. Watson is not here. Young is here. Zweiback is here. Lawrence is not here. Wilson is not here. Did I miss anybody? All right, the first word for attendance, pay attention here because it's going to be a little bit different. Since today is Halloween, you can either tell me what you're going to dress up as for Halloween tonight, what you would dress up if you could dress up for Halloween tonight, the last time you dressed up for Halloween, what was it? Or your favorite Halloween costume, right? So there's a lot of choices there. And obviously, I'm not going to know if you're lying. So if you just say vampire, then you're going to get credit, right? What do I know? Full disclosure, it would just be a little bit suspicious if I ask questions like this and someone turns in all the same answers. Like, oh, okay. See. But anyway, that out of the way, let's jump back into chapter 9, the last chapter for this exam 3. Last thing we were talking about was animal viruses. Talked about how there could be RNA viruses or DNA viruses. And then we left off with this. And this is just the basic schematic of how generally an animal virus would work. They're all a little bit different, obviously, but this is generally how they would work. So you've already seen the pictures of the viruses. You know that these animal viruses have a protein box with genetic material on the inside, like all viruses do. But you know that they also have this membrane with spikes on the outside that helps them merge into this cellular membrane, right? So it's merging into the cell. This viral material is entering the cell now, right? So then this protein cover disintegrates, so to speak. It's uncoated. Speaking of which, your textbook and I don't explain how that works, so you can look that up for extra credit. Like what happens? What does this job? What's uncoating the protein so we can get to the genetic material? But anyway, it gets uncoated. That leaves the viral RNA. Again, assuming this is an RNA virus, not a DNA. So here we go. We have this RNA virus. So then RNA is synthesized, right? So there's an enzyme that came along in this package that brings its own enzyme. That enzyme duplicates the RNA. And again, I'm not going into details. You can look that up for extra credit if you want. And let's scroll down to see a little bit more. All right, so we've duplicated this RNA. I think I said DNA earlier. I apologize. We duplicated the RNA, right? So now we have the original red copy and we have the duplicated purple copy. One copy goes over here. And again, it's going to be copied and copied and copied, right? So we have this new viral genome. And that copy will be used to be inserted into the new virus. And then, of course, this other copy comes over here, and it's the one that's translated, right? That's the one that says, all right, here's how you build this particular protein for this virus. Here's how you build this particular protein for this virus, right? So this is the one that's translated. And your cell just reads all that information, right? The ribosomes read the information. They build the proteins. And then those proteins that are built, plus this new RNA, they all come together to build a new virus. And then, again, eventually, like you've seen in the previous pictures, it's not just one virus doing this, right? There's a bunch. And eventually there's so many that the cell explodes and then the virus eventually exits. So there you go. That's the general schematic, right? That's basically how it works. Here's a picture with all of it in one big picture. That's generally how it works. for an RNA virus. Any questions about that? All right. So if you download the PowerPoint, you can watch this video that shows how it works. Here's a picture of a particular virus from the old textbook, I think. This is a mumps virus. You can see the nuclear, or the membrane, right? The cellular envelope right there. You can see some protein spikes on the outside. Typical stuff, just like we've been showing. That one happens to be, like I said, the mumps virus, not that it matters. All right. It is worth noting, right? I just showed you this picture of this virus being, this RNA virus in that last example, being duplicated inside the cytoplasm, right? But not all of them do that in the cytoplasm. Sometimes they happen inside the actual nucleus, which, as you might imagine, is usually a little bit worse, right? So for example, and you don't need to know these examples for the exam, but, excuse me, there we go. Herpes virus, which includes chickenpox, shingles, cold sores, genital herpes, all that stuff falls under herpes viruses. These are enveloped DNA viruses, and they reproduce in the cell's nucleus. And they get their envelope from the cell's nuclear envelope, right, the cell's nuclear membrane. So again, as you can imagine, this might be a little bit worse. If it's happening inside your nucleus, where your one genome is, right, that could be dangerous. And we're going to talk about why here in a second. Are there any questions so far? All right, so for example, talking about the herpes virus. The DNA in that herpes virus remains behind in the nucleus of certain nerve cells. So in other words, it incorporates itself into your genome. You got this this virus that attacks your cell. It inserts its DNA into your cell, which makes its way into your nucleus, right? So therefore, it is now part of your it's now part of your genome. And it can remain dormant in your genome until something triggers it, right? So maybe you get sick, maybe there's some stress, something happens and it triggers it. And then that's when you have these flare-ups. So I'm sure you've heard like people would get like cold sores every once in a while. They don't usually always have cold sores. I mean, it's there, it's dormant. They always have the ability to have cold sores, but it's not like they always have cold sores. 75%, not that you need to know that number. Likewise, more than 20% of adults have herpes simplex 2, which is genital herpes. And again, that's the type where you don't sound like you're always having sores, but sometimes things trigger it and it flares out. So again, this is just an example. I'm not going to ask you any questions about herpes virus. The point is that sometimes viruses are a little bit more, the word they use for herpes is they call it the gift that keeps on giving, right? And that's why it gets into your genome and it's there forever. And again, with the nerve cells, right? Nerve cells aren't replaced like a lot of other cells are. So once it's there, it's there. For extra credit, you could look this up. So I think this was written in 2016. That's almost a decade old. Of course, it's pretty vague. It just says over 75% and over 20%. But if you want to, for extra credit, you could look up what's the new stats. What percentage of people in the U.S. have herpes simplex 1 or herpes simplex 2? And that actually doesn't say the U.S. So wherever you want to look it up, in the U.S., worldwide, pick a country, whatever, pick a state, pick a county, whatever you want. Just don't forget, if you do that for points, make sure you provide a link to your source so I can look it up and make sure what you wrote is correct. Anyway, any questions about this example of an animal virus? All right. Next word for attendance is doorbell. Today is also National Doorbell Day. Kind of makes sense. Because, back when I was a kid anyway, when you trick-or-treated, you would actually walk up and ring the doorbell and someone would come. Of course, these days, everybody seems to just be out there. It's easier for the kids. I like it. Anyway, so when we're talking about the damage a virus can do, it depends partially on how quickly the immune system responds. Obviously, so if there's a virus that's good at getting around the immune system, it's going to be a little bit more dangerous. Also, it depends on the ability of the tissue to repair itself. So again, whatever part that particular virus attacks, if it's good at healing itself, obviously that's a better thing. If it's something that's slow to heal, that's not a good thing. Or, as we're going to talk about later, think about the AIDS virus, HIV. It attacks your immune system, and your immune system is the things that protect you from well, from viruses, right? So when you think about it that way, it's very bad, right? Because you're attacking the thing that's supposed to protect you. But anyway, that being said, this is why most people fully recover from colds, right? Because colds attack the respiratory tract, and the respiratory tract is generally good at repairing itself. Of course, you could probably, off the top of your head right now, think of some other things like, wait a minute, I can think of other bad viruses that also attack your respiratory tract, and they're pretty bad, But anyway, you can look that up for independent work if you want. I mean, at your credit. And I'll say sometimes people don't fully recover from colds, right? If someone's immunocompromised, they might not fully recover. And like I mentioned the other day, the cold virus is a coronavirus. And as we all probably know, the virus that causes COVID-19 is also a coronavirus. So they're very closely related. So anyway, the example here, again, the idea here is that some viruses are worse than others, and it depends on how quickly your immune system can fight the infection, and also depends on what it's attacking. So the one example we gave for, hey, it's not that bad, is a common cold, right? A lot of us get common cold, no big deal. The flip side of that would be something like the polio virus, right? The polio virus attacks nerve cells. Nerve cells are usually not replaceable. So that makes it more permanent, right? If you damage that, that's it. It's permanent damage. So with the polio virus, the only medical option is to prevent it completely with things like, with vaccines. Now, I'm sure you guys have seen people in iron lungs, like people who have polio, right? So there are medical interventions to keep them alive once they get it, but they're not going to be cured of polio. They're just going to be kept alive. So the only way to prevent polio, the best way is with vaccines. Anyway, those are just examples. I'm not going to ask you any questions about the common cold versus the polio virus. Are there any questions about it? All right. The old textbook had something, I mentioned this before, every chapter they had something called the process of science, where they took the concept of that particular chapter and they brought in like a current event to talk about it, or a real life study. So I'm not going to go through this. I'm not going to ask you any questions about it, but you can look into it if you want. It's all about the West Nile virus. But I would recommend reading it. It is good to go through this process, right? The scientific method. That's the whole one of the purposes of this class, right? Is to learn about the scientific method, critical thinking, scientific thinking. All right, there we go. We're almost done already. So now that we're done talking about animal viruses, we're going to talk about a very specific animal virus called HIV. Right? This is a very interesting virus, and you'll see why. But there may be questions about this one. So all the other ones were just examples. HIV is very specific. It is very different in certain ways. So for that reason, there might be questions about HIV on the exam. Make sure you pay attention to this portion. So the disease is called AIDS, acquired immunodeficiency syndrome. That's the name of the disease. The virus is called HIV, human immunodeficiency virus. A lot of people get that confused. Not that it's that important, but there's the name of the disease and there's the name of the virus. So for example, again, COVID-19 is not the name of the virus, that's just the name of the disease. Anyway, what makes this interesting is this thing is something called a retrovirus. All retroviruses are RNA viruses, but they reproduce with a DNA molecule. So think about that. That should be odd to you if you're paying any attention. I know it's early in the morning, but if you paid attention previously and you understand how we go from DNA to RNA to protein, and if you're paying attention right now, what I just said to you should be very strange. I'm saying it's a DNA virus, an RNA virus, but it reproduces with the DNA molecule. So how would that work, right? This is the reverse of the usual DNA to RNA to protein flow, right? In this case, it goes backwards. It goes from RNA to DNA. How does it do it? And this will almost definitely be a test question if we talk about AIDS or HIV. It does it with an enzyme called reverse transcriptase, and that should be easy to remember in the multiple choice exam. It's not like you just need to come up with it out of nowhere. You just need to pick it out of a list, and it's in the name, right? Reverse transcriptase. It's the thing, transcription is going from DNA to RNA, so the reverse of transcription is going from RNA to DNA, so obviously it would make sense that the enzyme is called reverse transcriptase. There's more to talk about with AIDS, but are there any questions so far? All right, here's a picture of here's a drawing, a very simplified drawing from the old textbook. It should look like a lot of what the other ones that I've shown you look like. It's an animal virus, so you still have that membrane with protein spikes on the outside. It's a virus, so you still have like a capsule of proteins with genetic material inside. All that's the same. In this case, it happens to be RNA, not DNA. Here's the new thing, those little green balls. That's the reverse transcriptase. That's the thing that is reading the RNA and making DNA out of it, right? It's doing the reverse of transcription. Here's a picture of it in action. So here we have the viral RNA. Again, reverse transcriptase reads the RNA and makes a strand of DNA from it. Then something, and you can look this up for extra credit too, because we don't get into it. I'm not going to ask you any questions about it. Then something takes that single DNA strand and turns it into a double DNA strand. And of course, as you've learned before, the normal way of doing that is through DNA polymerase. So the question that I'm asking you is it DNA polymerase is doing this again, or is this something else? Because notice, remember, this is a good review for the exam. And a little tangent here. Remember, when we were just talking about S phase of interphase, when your chromosomes are duplicated, we talked about that earlier in the chapter. We said that DNA polymerase did that, right? And that all happened inside the nucleus. Now I'm saying this part's happening outside of the nucleus. So the question is, again, what is doing it? What is taking this single strand of DNA and duplicating it? Is it DNA polymerase? Is it some other enzyme? Did the enzyme come with the HIV virus? Sort of like this reverse transcriptase enzyme did. Anyway, once we have this double-stranded DNA, it then enters the nucleus. That's another extra credit question for you if you want. How does that happen? Right? Shouldn't be there would be something safeguarded. How does this DNA get into your nucleus? Anyway, once it gets in there, this is where it gets even trickier, even crazier. It inserts itself into your chromosomes. Right? It is now a part of your genome. Like you can see right there. And this, they call that a provirus. That virus has inserted itself into your chromosomes, which is yet another question for extra credit. How does that happen? What's the name of that process? Which enzymes are involved in taking that HIV genome or genetic information and putting it into your chromosomes? And of course, you have to wonder too, if it's not, or if it's random and it's just putting it in any old chromosome, what are the chances that it's going to disrupt an important gene, right? If you have a gene, like if this is a strand of DNA here, and this is a particular gene from here to here, and that's a really important gene, right? And then this comes in and inserts itself right here. That gene is no longer functioning, right? Because you've inserted something in the middle of it. So the question is then, again, how damaging could that be? How often does that happen? Does it happen at all? All things you can look up for extra credit. But anyway, again, this viral DNA is then inserted into your DNA. And as you know from previous chapters and the chapters that you're going to need to know for this upcoming exam, exam 3, it goes through mitosis. And it doesn't know any better, right? DNA polymerase, when it goes through mitosis, it's going to read this whole chromosome and just copy it, including this new provirus, right? The DNA polymerase doesn't know it's a provirus. It's just going to do it. So now every time this cell copies, and then that cell copies, and so on and so forth, that's what mitosis goes on and on. It's going to keep copying that copy of the virus, right? It's almost like the lysogenic cycle when we talked about lytic cycle versus lysogenic cycle with bacteriophages, right? The lytic cycle, the virus attacked the bacteria that just killed it right away. The lysogenic cycle, the bacteria would enter itself into genome of the bacteria, and then every time it duplicated, it got copied, right? So this is, in a sense, the same as the lysogenic cycle. But anyway, eventually, just like in the lysogenic cycle, this viral DNA is transcribed, right? So just like it normally would be, RNA polymerase reads the gene, starts at the promoter, ends at the terminator. Now we have this molecule of, well, it should be mRNA, right? The mRNA, it gets capped and tailed, and it gets ready, it leaves the nucleus, and then just like usual, that gets translated, right? That viral RNA is translated. So now we have the proteins that we need to make the new virus. We have the RNA ready to be inserted into the new virus. All the parts are put together, and then finally it leaves and goes on to infect another cell. Whew. But again, there's a lot of stuff I didn't tell you that you can look up for information. So obviously you already know how this RNA is translated into protein. You already know about that, right? You know about initiation, elongation, termination. You know how that works. But what is putting all this together? So now we have the viral RNA, we have all these viral proteins, but what actually assembles them and turns them into one new virus? All that stuff you can look up for extra credit. Any questions so far? Okay. So this is oh yeah, never mind. If you download the PowerPoint, this is a little video, a little animation you can watch that basically shows everything I just talked about. It's the HIV reproductive cycle. So take a look at it if you want. Now, why is HIV virus so bad? Like I mentioned earlier, now it's just in writing. HIV kills white blood cells, right? The white blood cells are important for your immune system. It's like the Marine Corps of your immune system. Those are things do well, part of it. It's a thing doing the killing. When this was written, there was no cure for HIV. Of course, things are changing. You can look into that for extra credit if you want. Again, don't forget to use your sources, include links to your sources if you do that. But there are two categories of drugs that slow the HIV virus. Both of them basically work the same way, if you get really broad here, by interfering with the reproduction of the virus. So it isn't as if it goes in there. It's not like you take these drugs and then your immune system is better at killing the virus, right? Or anything like that. It's not like that. This is actually interfering with the reproduction of the virus. So it's not necessarily killing the virus, one specific virus, right? It's preventing it from reproducing. So in a sense, you could say it's killing the population of viruses by preventing them from reproducing, but it doesn't go in and kill like one individual virus. So the first type, again, there's two categories. The first type inhibits something called a protease enzyme, and these protease enzymes help produce those HIV proteins, which is kind of a hint at some of the stuff I was asking you about earlier that I said you could look up for extra credit. Like, what enzyme is doing this? What enzyme is doing that? So now you have one word you can put to it to help you look into it if you want for extra credit. Protease enzymes. So again, those are the things that are helping produce the proteins needed for HIV. And the second type, and there's two categories. The second category inhibits that reverse transcriptase. So remember, reverse transcriptase is that enzyme that reads the RNA molecule from HIV and then makes a strand of DNA from it, right? So this thing inhibits that enzyme. Remember, we learned a long time ago about inhibitors, right? So now they're coming back. This thing inhibits that enzyme that does that job. And this is another great example of something I've been talking about all semester long, this relationship between form and function, right? How the shape dictates or indicates how it works. Same thing here. Here's an example. I'm not going to ask you any questions about this, but the way this particular drug works is AZT. Here you can see this is what a T nucleotide looks like. You know what they look like, A's, T's, C's, and G's. It happens to be a T. This is what a normal one looks like. Now, when you take this medication, now you're going to have these in your system. But instead of having a T, you have this molecule called AZT. And it's very similar. Can anybody see the difference? I'm not going to wait too long, but let's see if anybody can see the difference. It's like one of those games where like you have two photos that are identical. You have to pick out the differences. There's only, well, I think there's only one. There's more. Yeah, you're right. There's more than, yeah, I should have known that anyway. That's actually important, the fact that there's two. So I feel stupid that I only said one. So does anybody see, I guess you can see the two differences? Great job. When you send your words, if you remember, remind me that you said that so you can get a little extra credit. So see here, we have CH2, right? That's ready to bond to something else. Here we have a CH2 that's already bonded to that OH. Now you don't need to know the chemistry of it, but the point here is there's a solid line here saying this thing is bonded to that thing. This thing has a dashed line because that's basically indicating, hey, this could bond to something else. This OH, well, that's a bad example too. But anyway, because this OH could also bond to something else. As you know, there's the sugar phosphate backbone, right? Point here is this thing is ready to bond to another thing, which is what you need. If you're building a DNA molecule, you need to bond these A's, C's, T's, and G's together, right? You need to bond them. This thing does not do that. This thing will not bond. It's like it ends the process. So once your reverse transcriptase brings in this instead of a T, that's the end of the process, right? Because then it cannot covalently bond anything else to it. So that's the end of the road. And that's how that works. But again, that's just an example. I'm not going to ask you any questions about it. Do you have any questions about it? That's it for HIV. Like I said, we're going to have an early day. So the last portion of this chapter is talking about prions. Again, this is a non-cellular infectious agent. So it's not alive, but it's still infectious. And I think we brought this up before. When I first introduced you to chapter 2 to proteins, I think we mentioned prions at that point. I know we've talked about it at least briefly before, but here we go again. Prions are infectious proteins. You need to know that for the exam. They are infectious proteins. They always cause brain diseases. Next word for attendance, breadstick. For some reason, today is also National Breadstick Day. Anyway, so you might be thinking, how are proteins infectious? How are they causing diseases? Well, the reason is because they are a misfolded form of a normal brain protein. So you have a normal protein that belongs in your brain, and then it gets misfolded. And remember, when you learned about proteins, it's all about the shape, right? If you mess up the shape, you mess up the function, especially with proteins. But imagine it's probably in your brain, you might imagine it's even more important because your brain is a very important part, right? So what happens is it gets into a cell that has normal protein, and then just by being around it, it converts that normal protein to a misfolded version of the protein. And then they all start clumping together. And because of that, basically you have holes in your brain. But for the exam, basically what you need to know is a prion is an infectious misfolded protein that basically attacks your brain. I'm going to give you some examples. I'm definitely not going to ask you about these examples, but you can look any of them up for extra credit, or you can look up to see if there's any other prions that I didn't list. But there's something called scrappy in sheep. If you're around here and you hunt, you're probably aware of this one, chronic wasting disease. You can get that in deer and elk. You get mad cow disease, obviously in cows. That one's the one I'm familiar with because when I was a kid, that was big on the news. So it sticks out with me. There's something called Creutzfeldt-Jakob disease. So that's one that attacks humans. It's incurable and inevitably fatal. And then again, I think I mentioned this before, but there's something called Kuru, and you can look that up for extra credit if you want to. Kuru is the interesting one because that's the one that was caused by cannibalism or spread by cannibalism. So there's humans eating other humans' brains, and it spread. And there you go. So for extra credit, like I said, look anything up about it. Is it still around? How prevalent is it? So are there still people eating other people's brains? If not, how's it getting spread? And again, are there other versions? Right? I know this is a short list of prion diseases, but a very short list of animals too. Sheep, deer, elk, cow, humans. Think about all the other animals out there. Like, are there any other ones? Do fish get it? Like, think about big classes of animals or big, chunks. Do fish get it? Do birds get it? So far, everything here I've listed is a mammal. Anyway, any questions about that? All right. Again, this PowerPoint is based on the old textbook. The old textbook ended every chapter by bringing it into evolution. Like, all right, how does evolution fold into this? Like, what does this have to do with evolution? So you can read that if you want. I'm not going to ask you any questions about it. We are done early for the day. Please don't forget to include one question about the material presented in this lecture, even if it's not you asking me a question. It could just be you saying, like, hey, this would be a good quiz question or test question. That's it. Guys have a great Halloween. I'll see you on Monday. Monday. And I will be in my office and online until noon if you need me for office hours.